Welcome everyone, and as usually, we translate this into simultaneously into three languages, two languages. The talk is in English, but it's translated into Spanish and Chinese simultaneously. So it's important that you select an appropriate translation channel. And even if you're listening in English, do please select English channel. And the reason is that if there is a question, let's say in Spanish or Chinese, then the interpreter can interpret back into English and you can hear the question. If you do stay in the original channel, you will not hear the interpretations. So there is three channels to choose from. And the way you get the channel is you select a globe that looks like a globe on your bar for your Zoom, and on that globe says interpretation, and you select either English or Chinese or Spanish. Ursula, and then Yunfan, would you be able to say it also in Chinese what people need to do, okay? After Ursula. Yunfan. Yeah, Ursula?
Okay, I'm sorry. I was managing some.
That's okay.
So, buenos días a todos los hispanoparlantes, bueno, a todos, buenos días a todos y a todas, buenas tardes, buenas noches. Estamos en distintos horarios al mismo tiempo. Muchas gracias por unirse hoy a la charla de Giovanna Danies. Para nosotros es un honor tenerla aquí. Lo primero que les vamos a comentar es que esta charla será traducida o interpretada del inglés. La charla va a ser dada en inglés y será interpretada o traducida al chino mandarín y al español. De manera que los invitamos a seleccionar el canal correspondiente abajo de su pantalla en el icono del globo terráqueo, dice interpretación en castellano por lo menos, y ustedes pueden seleccionar audio original, inglés o chino o español, ¿ya? Aunque puedan querer escuchar la charla en inglés, les aconsejamos de todas maneras mantenerse o seleccionar el canal del inglés por si hubiera una persona que haga una pregunta en chino, por ejemplo. ¿Ya? De esa manera la van a poder escuchar en inglés o en español, etc. O sea, los invitamos a seleccionar el canal. ¿Ya? Bien, eso. Yo voy a estar a cargo de grabar la sesión en castellano, por lo tanto, de aquí en adelante me voy a ir al canal de castellano y voy a hablar en castellano. Ok, so, Yunfan.
你好，能听到我讲话吗？好的，接下来有请这个主讲人为我们讲座主旨内容，然后希望各位倾听的演讲者能够在演讲人作为主旨观点之后呢，提出来一些自己感兴趣的问题，也可以跟大家踊跃交流，在我们的对话框当中也可以，谢谢。
Thank you.
Great. Okay. And Ursula, would you like to invite our guests today to start the talk, please?
Por supuesto, gracias, Eric. Bueno, nuestra invitada del día de hoy nuestra invitada del día de hoy es Giovanna Danies. Yo creo que ya es una amiga nuestra, una amiga de nuestra escuela. Giovanna la conocí la conocí primero online, por supuesto, cuando la invité a escribir un artículo para Revista Base. Me llamó la atención mucho su trayectoria, que tiene que ver, Giovanna viene desde el ámbito de las ciencias, de hecho ella es bióloga, y se interesó por el diseño y diseñó una metodología muy interesante que combina diseño y ciencias para fomentar la vocación STEM en escuelas, ¿ya? Entonces, de alguna manera, su trayectoria está totalmente relacionada con el foco de Design Literacy, cuál es la enseñanza del diseño en distintos niveles, desde el kindergarten hasta el doctorado, como dice Lee. Giovanna es bióloga, es madre de Ernesto y Antonio y Elisa. Ella es bióloga, como les decía, tiene un doctorado en fitopatología de la Universidad de Cornell y un master degree en ciencias biológicas. Ella o está afiliada a la Universidad de los Andes de Bogotá y es profesora de la Escuela de Arquitectura y Diseño. Como les decía, es autora del libro Biodiseño en colegios, que está traducido como Biodesign for High School, y ganó el premio de L'Oréal y UNESCO para las mujeres en la ciencia. Además es directora del grupo Atarraya de la Universidad de los Andes, que justamente lo que busca es fortalecer las habilidades y vocaciones de STEM a nivel escolar en Colombia. Bueno, así que sin más preámbulos, quiero invitar a Giovanna. Giovanna, la pantalla es tuya. Adelante.
Thank you, Ursula, for the invitation. And today I've titled this talk, Biodesigning the Future, Reimagining STEAM Education in Schools. So I'm Giovanna Danies, and as Ursula mentioned, I'm a biologist and microbiologist with a PhD in plant pathology. Let's see. Okay. For over a decade, my research has focused on characterizing the causal agent of late blight disease, the same disease that triggered the Irish famine back in the mid-19th century. More than a century later, it continues to cause devastating losses in crops around the world. And during those 10 years, I had never asked myself a simple but intriguing question. If this pathogen is so successful, could it have enzymatic tools that might be useful for industrial processes, for example? Why? Because as a plant pathologist, our focus is usually on fighting diseases, and rarely do we look at pathogens through a broader systemic lens. But the world's most urgent challenges are complex. They demand interdisciplinary thinking. Even the Irish famine, for example, was not solely the result of a plant disease. It was also shaped by economic, political, and social factors. Understanding such complexity requires us to connect the dots between science, society, and design. In 2017, I was invited to co-teach an advanced design studio to help students prepare for the Biodesign Challenge, which is an international education program that brings together biotechnology, art, and design. It connects high schools and university students to a global community of leaders across academia, industry, and entrepreneurship, all exploring how biotech can transform our world responsibly and creatively. Today, I'm an associate professor in the School of Architecture and Design at the Universidad de los Andes in Bogotá, Colombia, where I work in the field of biodesign. The biodesign process, for those who have perhaps not heard of this term before, follows the principles of design thinking. So it has the same steps in the design thinking mindset. To spark creativity, I often begin asking my students to imagine dystopian futures, that is, negative futures, grounded in real data, futures that feel close and personal. And today we have AI tools that can help us in quick exercise make our students imagine. For example, today I'm in a city in Colombia called Manizales. So I can look for data and find what is Manizales going to be like in 50 years, right? Given our current projections, what will the temperature be in our city? If it's expected to rise, how might that change daily life? We might see new disease-carrying vectors, water scarcity, or the need for entirely new kinds of buildings. Once they've envisioned these scenarios, students explore existing or emerging biotechnologies that might help address these challenges. And also, there are AI tools that allow students to speak with research papers. So it's not that like before, when I was starting to teach biodesign, I had this gap when I was introducing research papers to design students. They were a little bit scared of how it was written, of not understanding. But today you can chat with AI and it can explain research paper. It can find other complementary information to help students understand the content. Other students take the opposite approach. So they don't go into the future thinking of negative futures, but they start with a fascinating biotechnology or organism and imagine how it could be applied to contemporary problems. So through these exercises, students quickly learn a key lesson, and the goal is not to fall in love with a solution, but with a problem itself. So us scientists, when we are working, we're usually doing hypotheses. From the beginning of our working process, we kind of know the solution we're expecting. But design students, I need them to forget about solutions. I need them to focus on the problem. When the problem is real, any solution they're going to arrive to is going to be fascinating. There are always multiple possible solutions, but the most meaningful ones are those that make sense for the community and context they're working in. I feel incredibly fortunate to do this work in Colombia, here in the northern part of South America, one of the most biodiverse countries on Earth. Our ecosystems are unique and are also our social challenges. Together, they offer a rich landscape of design opportunities that can be addressed through biotechnology and living systems. Today, we face interconnected global challenges, from social inequities and public health threats to the profound effects of climate change. Yet traditional education systems, which are often focused on theory and standardized testing, are not equipping students with the skills needed to respond creatively and collaboratively to these issues. As a plant pathologist, when I first began teaching, I knew exactly the first day of class what my students were going to learn the last day of class, because I had tests that evaluated content. Today, in my design courses, I know that students are going to come up with a question that is interesting and that it's relevant in their context, but I have no idea what they're going to work on. Science and technology can provide powerful tools, but their adoption depends on cultivating imagination, empathy, and interdisciplinary collaboration. And there's so much still to explore. In Colombia, the vast microbial diversity, for instance, holds untapped potential as future sources of dyes, scents, enzymes, energy, and anything you can imagine. Here, what you see in this image where I have written biodesign, it's microorganisms that can be found in our hands, in our cell phones, when you touch your desk. So they're all around. Now imagine what all of these microbes are able to do. You might be asking yourself, so, okay, I have said that we go into dystopian futures, negative futures, or we imagine biotechnologies that can be useful or adapted in future scenarios. But I will talk about two routes where students usually begin their process, and one is nature-driven biodesign, and the other one is the socially driven biodesign. So I'm going to have 3 examples to exemplify this. In the first example, students, this one that I'm going to explain, students became fascinated by one of Colombia's most unique and vital ecosystems, which we call El Páramo. It's a high-altitude wetland that supplies water to many of our main cities. While hiking through our national parks, they became intrigued by a particular moss known for its remarkable ability to absorb and retain large amounts of water. So initially, they imagined extracting proteins that are responsible for this absorption and using them as a sustainable alternative to the synthetic materials currently used, for example, in diapers. But while they were doing their research, they discovered a paper, which was, I think, a PhD thesis that completely shifted their focus. It was much more compelling problem to solve. They had read that on average, a person ingests the equivalent of 1 credit card's worth of microplastic every week. So they decided to tackle this issue instead. That research paper they had found, or that thesis they had found, mentioned that same mosque they were interested was able to trap microplastics from water. So what they did was design and build a water filter that could be easily adapted to existing faucets, especially in coastal regions of Colombia. In these areas, people often boil water for safety, but boiling does not remove microplastics. As a result, many are unknowingly consuming plastic through their water, the rice, and other foods. This project, with this project, the team received the overall prize at the Biodesign Challenge in 2022. A second example is nanofreeze, which began when students watched this video. So there were scientists adding a liquid to cool water, and mysteriously it froze immediately.
Okay, that's it. Push again.
So they first had no idea what the scientists were adding to water. So they conducted the research, and they found that it was a protein that it was produced initially by a bacteria called Pseudomonas syringae, which we can find worldwide. And it's a plant pathogen which has ice-nucleating proteins. So in nature, these proteins help bacteria freeze the liquid inside the tissues so that the bacteria can then spread throughout the plant. They further found that these same proteins are produced on an industrial scale and are used at ski resorts to create artificial snow when temperatures are too warm for not natural snowfall. So they apply the proteins in the air and they can freeze water at just 2 to 4 degrees Celsius. Inspired by this, the students proposed a new application, a biotech-based refrigerating system for transporting vaccine to rural areas in Colombia. And as designers, they went a step further, creating a backpack that healthcare workers could wear comfortably while traveling long distances, reducing the back pain they often caused by carrying heavy styrofoam boxes on one of the shoulders. So again, innovating is not necessarily inventing something from scratch, is finding or understanding a technology and finding a new application for that technology. Today, this is a patented nanobiotechnology developed in collaboration with a professor from our School of Engineering. And together, they enhance the protein's properties by combining it with nanoparticles. So I'm going to show you a video of one of the applications that NanoFreeze has today.
Having ice-cold beers 24/7 is a must for a brand like Corona. However, electricity costs account for up to 1/3 of our store's expenses. And refrigerators produce massive CO2 emissions. Cooling is expensive and is heating the planet. Until now, Corona, the leading brand committed to protecting our paradises, set out to change the rules of the industry. Introducing Corona Natural Freezers, powered by NanoFreeze. Bio-nanotechnology applied to our refrigerators, reducing energy consumption by up to 30%.
Desarrollamos un gel refrigerante 99% biodegradable que es capaz de mantener hasta por 5 horas temperaturas heladas sin necesidad de consumo energético.
But the innovation goes beyond the gel. We've encapsulated it in a system of adaptable panels and grids, paired with a thermostat sensor that monitors the gel's temperature. Once the system is installed, when the gel freezes, a smart controller shuts the cooler off. 5 hours later, as it begins to thaw, the cooler turns back on. This cycle repeats as many times needed. This is a major shift from a regular cooler, which runs consumption cycles 20 minutes per hour.
Nuestras tiendas son operadas por cerca de 8.500 empresarios independientes y hoy más o menos estamos identificando que el ahorro potencial puede estar cercano a un 30%.
Estamos ahorrando un 30% aproximadamente en el pago del recibo de la luz. Eso nos ayuda a nosotros a ahorrar y para invertir ese dinero en otras instancias.
Anteriormente mis recibos eran de 25,000 pesos y actualmente he pagado 14,000 pesos.
We have tested natural freezers in labs and stores across Mexico and Colombia, proving this is not just a business shift, but also an environmental breakthrough. There are more than 2.5 billion coolers around the world. Imagine what we could save if every cooler was a natural freezer. This technology is a revolution for Corona, for the industry, and for the planet.
And my third example is a socially driven biodesign project. This one is called Lixi Lab, which emerged in response to the challenges faced by a community living near a landfill outside of Bogota, which is Colombia's capital. In this area, the local water sources are heavily contaminated with heavy metals due to the leachates from this water site. As a result, the crops grown there accumulate dangerous levels of these metals, leading to significant crop loss and posing serious risks to both animals and human health. So by recognizing my students' interest in the issue, I connected them with a colleague from our university who had published research on a bacteria capable of absorbing metals on their surface. Using this knowledge, the students developed a portable bioremediation system that employs the bacterium Lisinobacillus ferricus to purify water contaminated with heavy metals. But their work went far beyond technology. The team also collaborated closely with the local community, conducting workshops and creating digital content to teach about bioremediation and the importance of environmental stewardship. So this is something that design does really well. When I worked as a plant pathology, I also learned of people's problems, but I learned them from research papers and I worked in the lab. And I rarely connect with the users. Once I went to the users, I found that they sometimes had solved or went around the problem I was researching, but they had other problems that I wasn't aware of. So working really close to the community is important, and transferring that technology to them is also very important. This project quickly gained international recognition. It won the award for Best Field Research at the Biodesign Challenge in 2022, second place at Cumulus Green 2022. It became a finalist in Prototype for Humanity, and it received the first place in the Transformative Research Challenge organized by the FAO in 2024. And today, Lixi Lab has evolved in a social venture dedicated to protecting biotechnology so it can be scaled responsibly, ensuring that the communities and others facing similar challenges have sustainable access to clean water. We now have only 5 years until 2030, the target date we originally set to achieve the Sustainable Development Goals. But today it is clear that being sustainable is no longer enough. We must go further. We must be regenerative, adding value to our planet and to society in every project we undertake. Now, when I go as a jury to some of the PhD theses, I see that their projects are becoming much more efficient because they have less people. I feel today we have to be responsible, and what are the people going to do? What are the new jobs going to be? How can we not only improve something in society, but also take care of the environment without obviously forgetting about the economic value? With this in mind, we created a book, The Biodesign for High Schools. It is a written guide for students, teachers, and anyone curious about exploring biodesign methodology. The book includes a set of cards that are designed to help define the challenge to be addressed. These cards combine sustainable development goals with different contexts and user perspectives, and they include thought-provoking questions to spark meaningful conversation with teams. Once you start working on biodesign, sometimes students are like, okay, where should I start? What problems should I work on? And of course, because these projects are in teams, everyone has their interests. So the idea of doing this brainstorming session is not to tell them what to do, but just to spark and start the conversation. It also offers recipes and links to videos, tutorials, so that anyone, anywhere can start exploring with growing biological materials. And the book is available both in Kindle and in print versions. So To close, I want to emphasize the importance of incorporating extracurricular activities in high schools to start this conversation early, to give students the opportunity to engage with real context-based challenges. Most extracurricular programs today focus on robotics, artificial intelligence, programming, sports, and arts. These are all valuable areas, but there are still very few opportunities for students to explore biotechnology or design thinking. Imagine beginning a class with a wow moment. For example, showing students the isonucleating protein effect to open a discussion on what proteins are and the many applications they have. From creating artificial snow to developing new forms of refrigeration, for example. We can talk about biomimicry, which is the science of understanding that nature is our greatest teacher. It has billions in the planet has billions of years, and it has been facing one of the most pressing challenges us humans have. For example, lack of water in deserts. And the amazing thing about nature is that it doesn't have a single solution. It is redundant in solutions. Plants have different solutions, microbes have other solutions, animals have other solutions. So we can learn about nature solutions to create things that behave like nature. Now imagine designing kits to take genetic engineering to high schools. Or by doing bioplastics, we can talk about chemistry, we can talk about physics, we can talk about microbiology. And about fermentation, we can also mix a lot of interdisciplinary topics. Or introducing them to living materials by fermentation, like the bacterial cellulose, which teach patience, care, and an understanding that biological systems require time, nurture, and adaptation to their environments. By reconnecting students with nature, we don't only inspire curiosity, we help them reflect, appreciate, and value the living world that sustains. Thank you. So I'm happy to answer any questions.
Thank you so much, Giovanna, for really interesting talk. And I have questions, but I'll go with Jorge. Jorge, would you like to come in and ask a question, please?
The difference
yes,
between...
Thank you very much. Thank you, Giovanna, for such an inspiring seminar, honestly, I have to say. And my question is regarding what is the main difference between biomimicry and biodesign? Is it the moment of the question you want to answer? Or, for example, if I want to, fight or to save water, for example, because we have a water scarce problem, whatever, do I have to go for biodesign or biomimicry? I have to ask nature how nature has solved this problem before, or I have to find a way in biodesign to design something artificial. What is the difference between biomimicry and biodesign? That's my main question. Thank you.
Mm-hmm. Thank you, Jorge, for that question. So as I see it, biodesign includes biomimicry, but biomimicry itself it's a whole field. So not all of biodesign is biomimicry, right? And biomimicry, as you mentioned, is when we have a challenge and we want to see how nature has solved that challenge before. If we want to explore strategies that come from nature, then we look into the biomimicry process. But there are other things, for example, biotechnology on its own. So a biotechnological process, such as like synthetic engineering or fermentation, or when we want to create biological materials, that route is not necessarily biomimicry. For example, if we want to create nanofreeze, for example, you could have approached it through biomimicry if the students would have asked, how does nature freeze, right? But their approach did not ask nature at the beginning. It just looked for biotechnologies and then found applications. So it is related, but it's different because biomimicry itself has its own tools on how to find different ways how nature responds to challenges. And you can be interested either in the function or in the strategy. Did I answer the question, Jorge?
Yes, thank you. Because, for example, if you want to produce insulin, let's say, that's a bioprocess, and we are using biotechnology.
Exactly. But it's not biomimicry.
It's not biomimicry.
Exactly.
We just take advantage of a property of genetic engineering in this case.
Exactly.
But then biodesign is to search for functions that could help another problem, but it's already in the property of the molecules or on the structure, cellular structure, let's say.
Yeah, and Jorge touched something I always talk in class, and it's genetic engineering. Genetic engineering is always a hot topic. I love because it creates debates and discussions, and it allows me to kind of share my impression that technologies per se are not bad, are not good or bad. They're just technology. How we apply them might be good or might be bad. But no one is against insulin, for example. And very few people know insulin, human insulin today is a genetic engineering process. It's bacteria that have the human insulin inserted in their DNA so that when they reproduce, they produce the protein, which is then extracted. And it's the protein that people with diabetes buy, right? But when we talk about genetic engineering crops, for example, then people are aware of it. Oh, I don't know if I want to use it. I don't know if it's going to be bad for me. And it's not going to be bad, but sometimes I understand there's also issues of equity. Can small farmers access the technology, or can the fact that we have a gene that is resistant to herbicides, does that incentivize that people add much more herbicide than they should? So there's a lot of questions that come around the technologies, but the technology itself is not good or bad. Thank you, Jorge.
Okay, next one we have a Clive. Clive, can you come in and ask your question, please?
Hi, Giovanna. That was fantastic. I was very interested in your opening slides about the potato famine in Ireland and then the devastation of crops that are ongoing. And you mentioned kind of the social construct, and then there's a kind of disease. In human beings, of course, we have vaccines, which are a kind of a minimized dose of the thing that potentially could kill you. Is there an equivalent in the world of plants and crops where, I mean, you mentioned kind of genetic engineering, we can engineer crops to be resistant, but is there a kind of direct equivalence between vaccinating crops against disease and some kind of equivalent mechanism being applied to industrially produced crops?
Yeah, there's like a strategy to genetically transform crops using a bacteria that what it does, it's create tumors in plants. So what scientists have done is that they remove the genes that create the tumors, but they leave the gene that the bacteria has to insert itself into the plant's DNA. And then what we do in a lab is that we engineer the bacteria so that it includes that gene that enters into the plant's DNA, but we also include other genes that we want. So we can include, for example, genes that will give the plant a higher nutritional value, or genes that will make the plant resistant to certain viruses, things like that, right? So then once the bacteria, which is able to infect the plant and insert its DNA into the plant's DNA, then when the plant's DNA is reproducing, it will reproduce the genes that we have inserted. So that's kind of what I think would be an equivalent for vaccines in plants.
The interesting thing about that, of course, is human beings have selected for the kind of properties you mentioned, kind of more wheat, nutrition, and all sorts of things. And by selectively breeding, you can get the kind of crops you require. But is the process you've described is an accelerative process? Is that something that could result in, instead of years and years of development, is that something that could accelerate the process of resistance to blight and disease in plants?
Yeah, it's like, so for example, I'm going to do an analogy with cows.
Okay.
So when you have a dairy farm, you have your cow, and then you see what cow produces most milk and it reproduces very well, and you breed that cow. And then perhaps you have a cow that doesn't give you enough milk, the one you're expecting, so you stop reproducing that cow, right? So you are selecting with time cows that are very efficient at producing milk.
Yeah.
So it's a very short, I mean, it's a process of waiting for the cow to have its calf, and it takes 9 months and like that. But if with genetic engineering, you can, or by a genetic process, you could see the DNA, because today we have access to sequencing DNA of every organism, right? And understanding the genetic code. So if we know what the genes are for selecting the characteristic we want, so for example, resistance or a nutritional value or any property, for example, it could be color, it could be sweetness, it could be any trait. If we know what trait we're looking for, we can see the seeds, then genotype the seeds, and only plant those that have the traits we're interested in. And that will be like, it would be much more efficient than just breeding and growing and waiting for the traits to develop themselves. So yeah.
There's a follow-up question to that, Giovanna. In terms of kind of ethics, what are the ethical implications of that? Because we, you have big companies who may control, the genetic reproduction of, seeds and plants that aren't available to kind of small-scale growers, and so is there an ethical consideration to all this?
There is, and I think that's where the debate starts, because some people have access and others don't. I believe that technology, with time, it becomes more cheaper to do it, so more people will have access to it, but it's something we definitely have to talk. With this topic, I always talk to my students about human genetics. So for example, there's something that is happening today, and is that because since there's in vitro fertilization in humans, people can select the gender of their baby. So I know of families that, for example, have 2 boys and they want a 3rd child, but they don't want a 3rd child. They want a girl. So they're not going to risk it with 50-50 to see if a girl is going to be born. So what they do, even if they can naturally have a baby, what they do is artificial fertilization to choose the embryos that are XX chromosome to have a girl, right? And then they implant that and they have a girl. So that you can do today. But then imagine what the society is going to be like. There's some people who can access the technology and can decide if they're going to have boys and girls, and there's other people who cannot decide that. And then what's the step from designing gender to designing eye color, to designing the type of hair that person is going to have, to designing other traits, right? So this is something I love to talk in my classes, because we start talking about the fact that a technology allows you to do things. It doesn't mean we can't do it. It doesn't mean it's wrong, but we have to really anticipate of what's going to happen if this is universal. What are the risks of us selecting how us humans should look? There has to be something about being diverse that allows us to keep living safely and advancing in the world, right? And then there's going to be discrimination by, oh, you were created, you are natural, right? Perhaps that's going to happen. But it doesn't mean we have to stop researching, or that this technology is bad. It can help in certain situations. If a baby is going to be born, for example, with diabetes or with an intolerance, and if you could change that, it's going to help that baby have like better conditions in life, right? So the well-being of the baby is going to be best. But there's a really thin line between what is allowed and what isn't allowed, right? And today we're there. Some people are allowed to choose things and others are not. So it's interesting, and we need to start the conversation.
That's been absolutely fascinating, Giovanna. Thank you so much. I've enjoyed your presentation so much, and I certainly learned a lot. And and I appreciate the time you're taking to take these kind of questions because, I mean, ethics is a minefield for most disciplines, isn't it? So, thank you very much.
If you're interested, I have so we have created a set of questions in for all of the design thinking process. So in all of this, okay, I stopped sharing screen, but let me go back here. So where's the presentation? Oh, here it is. So in every step of the design thinking process, students have some ethical considerations they have to reflect upon. Mm-hmm. So when they're exploring, when they're choosing what project they're going to work on, why am I going to choose this topic and not that? Where can I help most people? Where can I do the greatest impact? When I'm exploring with people, am I considering like what things they can be susceptible to what I'm saying or talking about, right? So they have to design their questionnaires thinking about others, how they can be heard, or also hear people, because scientists, we usually, as I mentioned, we are in a lab, our lab works on certain things and we continue working on that. But what I really admire from designers is that they not only work on the theory, they work with the people they are designing for. And that is super important because when I taught design, this course, the first years I taught it, I remember my students in Colombia wanting to address things like nutrition in Africa, but they had never been in Africa. They had never spoken with a person in Africa, so they didn't understand their context. And then whatever solution they're going to come up with is not necessarily going to be adopted. They don't know what that culture eats, why they eat it. Do they have access to that technology or not? Do they like the flavor of what they're proposing? They don't know that. So they're going to have a solution, but the solution is not going to solve the problem. So that's every step of the design thinking process has its own ethical considerations that enhance the projects and make them much more relevant.
Okay, thank you so much. Can I go to Lindsay? Lindsay, would you like to ask your question, please?
Hi. So my question is, could you share more details about how did you collaborate with the local communities, like on the socially driven projects? Like how did you go into the community and how to communicate, and what's the problems you will meet? Yeah.
So that's a very good question. I always ask students to work on problems with communities they are close to. So they have to have access to the community somehow. Either they're part of the community, or they live close to the community, or they have someone who works inside of that community, so that they can first enter into the community. Sometimes they do not have, but they begin exploring. So for example, I've had students who want to change the process, how leather is created, because there's a lot of heavy metals that are applied in the process of creating leather. And that is contaminating a lot of water sources. And when they go to these places and they start interviewing people, people become a little bit scared because they feel like the students are, I don't know, the police or something that is wants to know their process or wants to judge them. So it's hard to go into that community. And that happens. And some students, even if it's hard, they try and they ask and they go and they explore in different ways, right? But it's easier to work with a community that you're close to, or that you know someone who is inside that community, and make them part of your group. So my invitation to do this type of things is to incorporate into the team someone who is from the community.
Great, thank you.
You're welcome.
And I also got another question. So So all of the projects you shared, it's really in a professional level. So are the students being trained both the engineering skills and design skills together in the course?
So students are designers. I have taught this course in different ways. So I have taught it by co-teaching with engineering professors, with science professors, and myself. Today I teach it on my own, and it's only design students. So it's been funny because at the beginning, students asked, they said, we feel the course would be better if we had scientists in our class. But then the first time I had scientists in our class, it was a really weird experience because scientists do not understand the project-based teaching strategies. For scientists, our classes are always content-based. So they expect teachers to come to class and always give them content. So they feel that this project-based teaching strategy is kind of superficial, like, what am I learning? What am I doing? And I'm like, look at your project. It's amazing. You're learning so much. But they feel they are not learning because they don't have like a proper PowerPoint lecture every time. So since that, when I learned that, I try to combine and I try to make them aware of their learning process. Today, you're exploring. Today, you're gaining experience and how to find or understand your problem from the community's perspective. Or today, you are doing a systems map. Why? Because we want to have a systemic view of the problem. Problems are not just a technological thing. They depend on the political issues, on social issues, economic issues. There's things that are positive, things that are negative, what people think, or some stereotypes that might exist. And they start seeing things they hadn't seen before. And then I also question them and I tell them, okay, you have this technology. Is that going to solve your problem? And then they say, okay, not necessarily, because people are not going to come or they don't have access to this. And they start understanding the value of that technology. So what I do as a professor is that when I see what my students are interested in, I connect them with experts in the field. And I always tell them, you are designers. You don't need to know everything. You need to understand what the other person is capable of doing. So also the first years I taught this, something was happening and it's that designers wanted to become biologists. So they went to the lab and they never left the lab. They started working on experiments and doing things, and they forgot about the big picture. They forgot about why am I doing what am I doing, and they started to focus on the details. And I had to remind them, you are designers, and that's amazing. And the idea of doing biodesign is kind of highlighting the design toolkit. Designers are not useful to do things pretty, to communicate things in an amazing way. They have very sophisticated tools they have developed in their careers to really understand problems, to really find solutions to those problems by reading literature, by interviewing people, by investigating, not only in a quantitative way, but also in a qualitative way. Us scientists are not experts in qualitative research. So that's something that adds a lot of value to all of these projects. And the technical things that you see is because students when you're interested in something, you learn about that thing, even if you're not in engineering, even if you're not a scientist. And that interdisciplinarity breaks and it becomes transdisciplinary. The designers start giving opinions about the science, the scientists start giving opinions about the design, and it all changes. But you always have to keep your lens from the discipline where you come, because that's highly valuable for the project.
Great, thanks so much. That's amazing to hear. And as a design student, it's especially cheerful to hear like all the project is done by designers, and a good way to collaborate with the scientists too. Thank you.
You're welcome.
Great. I think we have a last question, or we're getting close to the last question. I mean, there's more questions, but we're kind of running out of time. Anna, would you like to come in, please? Ask your question, Anna.
Hello. I'm very impressed about your work and attitude and all this stuff. But I have a question about this freezer. Thinking about history of food preservation, it could be big change. But what do you think? It is only Corona stuff, or maybe something which can be popular and instead of freezers we use?
Yeah, so that is the idea. The problem with technologies is always the adoption. Everyone is skeptical about, oh, does it really work? Somehow things that are biological, people have the sense that they are not as efficient. So when you go, for example, to my plant pathology world, when we talk about biological control, that means like microorganisms that help or avoid that bad microorganisms start growing. They think it's not as efficient as when you add synthetic chemicals, right? So it takes time, but that's the idea, that people have access to this. And what I feel, I always talk with my students, it's not replacing stuff, it's alternatives too. The real solutions are alternatives, and it's not charging the system with a lot of pressure. It's just, for example, you can have electricity, but if you have a combination of electricity and this bio-nanotechnology, then the bio-nanotechnology works, but when it's becoming inefficient, then the electricity comes in. But you reduce the usage of the electricity, like in a 30%, I think it's what they have tested right now. But where we are right now is that we have to do more research and publish research papers so that people understand that what this does and what this is capable of doing. And why we're doing things with Corona and other brands is because we need brands to talk about that technology and not us to talk about the technology. When brands are talking about the technology, people are going to believe more in this, right? Because, okay, they have tested it and they know it works. But if it's us saying that it works and we're doing this and that, some people think, oh, it's very romantic, it comes from the academia, but they don't understand how this works. So we need the users to start talking about the technology. But you're right, it's something that hopefully can go back into the initial purpose of the project, and is going into these rural areas that have less access to electricity, who have less access to for example, there's places in Colombia that are really, really hot. So how can you help cool environments with this? Also, that technology can continue developing. Can we have, for example, wearables with that technology? How would that work for athletes, for example, or people that are working under the sun and need this? So there's a lot to work on, and that's why we need more ideas, but also interdisciplinary teams that can make this happen.
And this is not very expensive, production of this?
It's not. So for example, for the extracurricular activity I mentioned at the end, I called Snowmax, which is the company that produces the proteins to release them in the ski resorts. Mm-hmm. And they sent me a package, so I'm now testing how to create the effect, the water effect. I'm experimenting that. What Wayne, the CEO, tells me is that it's hard for people believe that you're spreading bacteria in the air, and it's not that, right? These nucleating proteins are already in the air. The thing is that they are in very small concentrations in comparison when you add them by reproducing them. But the ice nucleating proteins are part of the water cycle. So it's also how we communicate this. And something he has shared with me is that it's very different when you tell people this is a bacteria or this is a microbe. Somehow microbes are like less bad, but bacteria are really bad. And there's a lot of like wording around things that it's amazing how it prevents people from using things. And unfortunately, there's a lot of greenwashing today. People don't have critical thinking. So when they buy because of what influencers are talking about, or ChatGPT is greenwashed. If you ask ChatGPT stuff, it's going to tell you what most people think, not what the real thing is, right? how efficient it is or how it really works. So that's something that makes this communication even more relevant.
Okay, thank you very much.
You're welcome, Anna.
Thank you so much. There were so many questions. There's more questions on the board, but we need to move on. Thank you, Giovanna.
You're welcome.
You know, for your presentation. I have a few questions myself, but maybe we do it another time. And again, thank you very much for engaging with us. Thank you for the people have engaged, you know, with your talk, which is important. And I'll go to Ursula, if she can take us off, please.
You can share my email if anyone has further questions. I'm happy to
Yeah, mute
answer.
you, Ursula.
I'm sorry.
Yeah, that's
I'm
okay.
going to share with you the email, Giovanna email. So it's okay, Giovanna? Okay.
Yes, please.
I'm sharing with you Giovanna email and also LinkedIn, so you can get connected with her. I'm speaking English because so the Chinese translator can translate this. So thank you, Giovanna, again, and well, and thank you all of you for joining today. And you are welcome to our next talk. Will be the 40th talk of our cycle or series, and the last one of this year, 2025. On November 25, Mauricio Mejia from the Design School of Arizona State University will discuss how professional and lay people engage in designing. And the registration are open now, so you can register from today. Eric is sharing the link for to the registration, and we will send also an email 1 week ahead to remember you. So I don't know, Liv, do you want to share a thought?
Well, I just want to say thank you to Giovanna. This was very interesting, and I'm really looking forward to hear more another time perhaps, and also for the next presentation with Mauricio later in November.
Sure.
Thank
you.
So
Thank you, everyone. You know, good night from Australia. Good morning to in Chile, and good afternoon in Norway and Poland, and good evening in China. So it was good to see everybody, and very good morning in Colombia. Yeah, very good morning. So and we'll see you again. We'll see you next in December. Okay, bye everyone.
Bye, thank you.
Bye. Muchas gracias, Giovanna. La Ale te manda un abrazo enorme y sus saludos, no pudo estar hoy día.
Bye. Muchas gracias.
Goodbye.
Chao, chao. Chao a todos. Gracias.
Hi, Linda.
Bye bye.